This is a production of Cornell University.
So I've been there a long time ago, just for a quick visit to Cornell. I think actually it was 26 years ago. I had the pleasure to visit Cornell, and in winter it's absolutely beautiful. I really enjoy being there. So today I will talk about some work that we are doing in our group at Bordeaux. And I will talk about the use of artificially induced genetic variability and show you some new work that we are doing. It's not unfinished stories. We are still working on that, but I wanted to share with you some results, the recent results that we got, and the strategy that we are currently using to identify the genes and alleles that are underlying phenotypic traits, quality traits in tomato. So, as we said before, I'm from Bordeaux. Bordeaux is a beautiful town, well known for the seaside, which is a wide seaside, and also, of course, for wine. And actually, in our lab, we are working on tomato, not on grape. And this is because, as I will show you later, tomato is a wonderful model for fleshy fruits. So this is our lab. We are a joint lab between INRA and Bordeaux University. And we will have the pleasure to welcome you at the end of this year to the SOL meeting in Bordeaux, the end of October. So, as I said before, tomato is a model fruit for many fleshy fruits, including grape, which is also a berry. And what we're interested in is mostly early fruit development before ripening, and especially the cell expansion stage, where most of the fruit weight is gained by the fruit. You have the largest increase in fruit weight during these stages, before, after the cell division and before the ripening. And what happens is that during the cell expansion stage, you have a huge increase in cell size. Cell size can go from 50 micron in some varieties to 1 millimeter length. So you can almost see the cell with the naked eyes. And what happens? We have a large increase in cell vacuole, increase in soluble content, sugars, organic acid, ions, and so on. You have also, and this is something that you don't find in grape, but that you have in tomato, you have a shift from mitotic cycle to endocycle, which is a simplified cycle, and a large increase in nucleus ploidy. And of course, you have a storage of starch that disappears during ripening, and membrane, cell wall, and cuticle biosynthesis to cope with this large increase in fruit size. So what we're interested in, and I will talk a little bit about that, is the mechanism that are involved in fruit development at the cell expansion stage, and the regulation, coordination of these processes. So I will talk about two strategies that we have been using in the last years. The first one going from identifying candidate genes and going to the phenotype. And this is, as I said before, unpublished results. We are still working on that, but I will show you how we try to tackle this question. So what you have done, this is the fruit. You have a carpel wall here, pericarp, And within the fruit, we have been interested in two expanding tissues between after cell division. Doesn't work anymore. Okay. After the cell division stage to the onset of ripening, so we have been looking at these cells here in the mesocarp, and you can see that you have a large increase in cell size. And the other tissues we have been looking at is tissues in the locular tissue in the gel, where you have also large increase in cell size, but these cells are clearly different and have a different fate. So we have been looking at that, doing metabolome analysis, transcript analysis. This has been done some time ago, so it was with microarray. And you can see that the two tissues, locular tissue and mesocarp tissue of the cells, are clearly different, have the same trajectory, but are clearly different. And for example, if you look at sucrose here, it mostly accumulates in the locular tissues, and the same for GABA and so on. So clearly, we have two tissues that are evolving during the cell expansion, but that are clearly also different. And we did the same for transcripts. What we were mostly interested in regulatory genes, and what we did is correlations between, for example, one metabolite here, again, one metabolite here, glutamine, transcripts, and so on. And we had several large number of pairwise correlation, and we went for looking at this correlation between metabolite gene, and then between genes and other genes and metabolites, and looking for hubs that are, in that case, genes with high number of correlations. And what we wanted to know is whether these hubs that show a strong regulation in the two tissues at 3 stages of development could be involved in what we are interested in, cell expansion or transition to other stages. So we draw a very basic correlation network, and we had several genes that were identified as hubs. Among them, what we had, and I think many people here know them quite well, much better than I do, berberine, and we know that this gene is very important for in triggering the onset of ripening. No, this is the same. This is a mutant, very important for ripening. AP2, we know that one homologue has been recently been shown to be regulating fruit ripening, and these two genes with no information. And so we were quite confident that these genes could be important for regulating fruit ripening, because the other candidates coming out were already identified as important regulators. So we have been looking into that. This is the two genes. One is mostly expressed during cell expansion and then decrease later. The other one mostly increasing after, at the end of this cell expansion. But what is quite interesting is that these genes, because we were working on cell expansion, these genes have their expression modified before the mature green stage. So during the cell expansion. So we have been using the tools available, using what we developed before, a fruit-specific promoter that is strongly expressed during the cell expansion stage, and this promoter drives expression specifically in the cells that are undergoing expansion in the fruit. This is in comparison 35 as a promoter. So what we did is overexpression, RNAi, no result. And then CREST technology, which basically allows us to have a kind of a dominant negative mutant for this basic factor. And what we have is these fruits are whiter, accumulate much less starch than the white type fruit, which may result in reduced accumulation of glucose, fructose later during ripening, more malate, increased firmness, which can be related to effect on fruit ripening, because what we saw is that this transgenic lines, different transgenic lines, we had a strong effect on fruit ripening. So we have been looking at that in detail. You can see the differences between BASIP and wild type. And you can clearly see that at BRCA onset of ripening plus 7 days, it's almost ripe, already ripe in wild type. You have ripe fruit after 15 days after BRCA stage. So we are at this point, we are working on that and working on different strategy to try to understand how this gene can be controlling fruit ripening. I won't go into the detail, but what I would like to insist on today in my talk today is the second strategy that we began to use to develop a few years ago, which is to go to identify mutants and to go from phenotype to the function of a gene. So one of the reasons, as I showed before, reverse genetics approach can be quite successful. We have tools now, and it was very basic at that time, but I think what is done now with RNA kinases, large correlation network, we can have very good insights upon the putative function of genes, but still we need to do plant transformation. We don't know whether it can have a phenotype or not, because it can be redundant and functions and so on. And what we wanted to develop, what we did, is to go for forward genetics approaches. So in tomato especially, I think there are wonderful resources developed with natural variability. But in most cases, what you need is to have large fields, greenhouses, human resources, and so on. And I like very much this picture from Danny Zamir, for example, with collecting fruits in Akko in the north of Israel. And I think most of the lab, at least ours, do not have this facility. What we have instead is growth chambers, greenhouses, and so on. So what we did, and we began that 10 or 15 years ago, is to develop artificially induced genetic diversity in tomato. And for that we choose the miniature cultivar microtome. And because it can be a very nice additional source of genetic diversity for functional studies of target gene when you have a candidate, for gene discovery, and I will show you how or sources of new alleles for breeding. And one interesting thing is that genetic background is identical between all mutants except for induced mutation. So what we chose is this miniature cultivar, because it's you can grow them in greenhouse year-round, high density, short cycle. They carry at least 2 recessive mutations. One is dwarf, but that affects parasinosteroid synthesis, not in the fruit, but in the vegetative tissues of the plant, and SP mutation, which affects, which control determinate growth. Sequence has been, genome has been sequenced recently, and so it's quite well adapted as a model cultivar for functional genomics in tomato. And something that can be quite interesting, when you use microtome, you cross it with one of these old parent, which is a moneymaker. This is F1, that looks like, that is very similar to the moneymaker parent. So what we did, and we did that several years ago now, we developed a mutant connection. We are currently using 8,500 M2 families for TINing, for identifying target genes, mutations in target genes. And we did at the same time phenotyping of 3,500 families. And this has been stored in a mutant database that we are using. What I have to say is that several collections in microtome are generated now, one being in Tsukuba University, and we have developed a joint lab for sharing these resources. So how can we use that? As I said before, we have been storing this phenotypic data in a database. You can search it for different keywords. And what we are currently doing, I won't talk about that today, but it's very, very interesting, screening for vitamin C mutants. And it has been done for sugar mutants, and you can have mutants with as much sugar than in grape, for example. And something that we are beginning soon is to screen for starch mutants. So this is just to show you, give you an idea of the diversity that you have. Actually, you have more than 35,000 different annotations, mutations that are phenotypic alterations that were annotated. So you can have a large diversity that you find in natural resources, in food chain, something that is very strange looking fruits. Color aspect, this one has been isolated, which is a cuticle mutant. And I will talk about that today. Is we are interested in fruit morphology, fruit size, and fruit morphology. And this is the work from Lucie Fernandez. And the other work I'm going to talk about, which is work from Johan Petit, who is there currently at Cornell for 2 weeks, is fruit surface aspect and cuticle. So what we are first trait I will focus on is fruit size. So fruit size depends on both cell number, carpel number. So this is determined mostly or in part before fruit set. And then after fruit set, you still have a cell division that will determine the number of cells. And you have also cell enlargement that will determine the size of the cell, and depends in tightly correlated in tomato to endoreduplication process, nucleus ploidy. And so what you see here is from the outer pericarp to the inside of fruit, and you have a cross section of a tomato pericarp. So what we have been looking at is mutants in with variation in fruit size and or variation in pericarp thickness. So this is how we do it. We screen search the database, for example, fruit, large fruit, looking at what we have in the database, including pictures, confirm that phenotypes by growing the plants. And this is what we can get. For example, this is Y-type fruit. You can see here, from here you have outer epidermis, inner epidermis, mesocarp here, very large cells. This mutant, which is a large fruit size mutant, you can see that you have more cells layer, but mostly larger cells. And this is linked to an increased, this is C value here, increase on the reduplication. So you have large cells with larger nucleus. In that case, what you have is thin pericarp, smaller cells, but in that case it is uncoupled from other reduplication, because you still have large nucleus but small cells. So to understand how the growth process proceeds in tomato, these mutants can be quite interesting. So what we have done is to work on these different mutants, characterize them, and so on. This is a principal component analysis. And for example, what we can see here is that we can have small fruits with small cells, but with both high ploidy and low ploidy. Low ploidy we are waiting for, but in that case we can have a small fruit, small cell mutant with high ploidy. Again, we are much interested in understanding how ploidy is controlling cell size and fruit size, and these are very nice tools. So this is what I said before. It can be very useful material. So we wanted, and we are focusing on several of these mutants. I will show you an example on this one, which is a thin pericarp mutant with low ploidy. And you can see here that the pericarp is very thinner than in white-type fruit. It has also, but we don't see it much here, a different shape. And what you have done, and I will go rapidly through it, what you have done is to begin to do a rough characterization of this mutant. So if you look at fruit rate in two different conditions, one with controlled fruit number, one with uncontrolled fruit number, in both cases in blue, fruit weight is lower. It has also an effect on fruit firmness. Here you have a Y-type here, and you have two different generations of this mutant, BC1, F2, and F3. I won't go into the details. These are the two colors, but anyway, firmness is higher in that mutant. And this is not due, because fruit size is very often dependent on seed number. And actually, this mutant has even more seeds than Y-type. So we have a small fruit mutant with high seed number, more dry matter, high firmness. And we have been looking at tissue level details. So what we did is to look at cross sections that you see here, Y-type and mutant, looking at pericarp thickness. Again, we can clearly see that pericarp thickness is different using a tomato analyzer developed by Esther van Knapp. We did the same for pericarp area. We clearly see that it's different. And we did also ploidy measurement, and we see here ploidy index. It's a low ploidy mutant. But when we looked, so it was quite interesting trying to understand why this pericarp thickness is smaller. We have been looking at cell tissue structure, different cells, and what we saw, and this is quite interesting, is that the mean cell area is much lower in the mutant. What you have is more or less, we say more or less because it's very difficult to be sure of that. It's more or less the same number of cell layers. But what we saw, and it's not clear from here, this is Y type. You look in the mesocarp, you have only large cells, except around the vessels. But in the mutants, what we saw is that these cells, some of these cells, instead of differentiating, expanding, and so on, are still undergoing cell division. So we still have, we believe, mitotic cycle, and this can last until ripening stage. So we clearly have a different control of probably the shift from a mitotic cycle to beyond the cycle in that mutant. And we have also looked at the different traits. The time until ripening is more or less the same, even shorter in that mutant. We have some defects which are not from cuticle defects, these brown spots. But what we clearly see also, and we couldn't separate this trait from this pericarp thickness trait, is that we have this difference in ripening, and we have uneven ripening in the mutant. And looking more in detail at ripening, what we could see is that actually it takes much more time after breaker stage to reach red ripe stage in the mutant. So what we believe is that we are still working on that. We have a mutant where you affect early fruit development, and at the same time you also affect ripening, so later stages of fruit development. So of course this is quite interesting, trying to look at these both processes, and we wanted to know which gene is underlying this trait or this trait. We have been looking, before that, we have been looking a bit more in detail. Tomato fruit normally accumulates soluble sugars. This is Y type. We accumulate soluble sugars. These are mutant lines, and clearly fruit composition is different. We don't accumulate glucose or fructose. And we have been looking, but these are very preliminary work, looking at the expression of known ripening regulators or ethylene-induced genes, and what you can see is that all the expression of all these genes is strongly delayed compared to Y-type fruit. So as I said before, what we wanted to do is to identify the mutation. So a strategy that we have been using is mapping by sequencing, taking advantage of the availability of a tomato genome since 2 years now, and current development of next generation sequencing technology. So one classical way for that is to outcross the mutant with, for example, Pampinifolium, which is another close but white tomato species. Problem that we have with that, working, for example, on fruit-sized mutant, is if you do that, crossing microtome with something that is quite different. What you will have in F2 segregating population is segregating for many, many different traits, including pericarp thickness, fruit size, and so on. So it can be very problematic. And this is the case for that. We saw that for Cuticle. When you have a very strong mutation, you can still phenotype population when it's more less severe mutation is very difficult. So what we did is to go for NGS mapping on the by bulk segregant analysis. So the idea is to cross the mutant, the yellow mutant, with white type microtome that doesn't carry mutation. In case, because this is a more simple case, monogenic recessive mutation, which was the case of the thin pericarp mutant, you can go to BC1, F2, and then you have a segregation between plants showing the white type phenotype and plants showing a mutant phenotype. You can bulk them, two bulks, white type-like and mutant-like, and then you can go for whole genome sequencing, what we did using Illumina, comparison with a reference genome. Nice thing with that is that the only source of genetic and phenotypic variability are the EMS mutation. These mutations can be used as SNP markers later, and I will show that this is very useful. So we did that. What you can wait for when you have a recessive mutation, causal mutation, homozygous here in yellow, and this is unlinked mutation in green. If you look at unlinked mutation in mutant bulk or white type bulk, the frequency will be 0.5. When you look at the mutation that you are interested in, the causal mutation, what you are waiting for is a frequency of 1 in the mutant bulk, but you have, because you took out all the plants with mutant phenotype, frequency is reduced in the wild type bulk. So we did that, and this is what I explained before. For example, unlinked mutation, if you look at a chromosome that doesn't carry mutation, you have a mean frequency around 0.5. If you look at one region in a chromosome that carries a mutation, but unlinked region, the same frequency. And then when you go close, very close to the causal mutation, frequency of the mutated allele increases, but that's the opposite in the mutant bulk. And what you are waiting for here is the causal mutation that has a frequency of 1. Or you can have a linked mutation that have also high frequency. So we did that. In that case, for example, we didn't have so many seeds, so we have been working on very small population. We have been working on 172 or 3 BC1 F2 individuals. We scored the different phenotypes. We had the expected segregation, where 25% had the thin pericarp phenotype that co-segregated with ripening phenotype. And then we constituted different bulks, for example, analysis of different traits, principal component analysis, bulk, and we sequence. And this is what we get after sequencing. So we did whole genome sequencing, 30 to 40x coverage. And this is the different chromosomes that do not carry the mutation here. You can see this is the sliding windows for EMS mutations. You can see that the curves are almost at the same level in the case of mutant and wild type bulk. But in the chromosome that carries the mutation, you have a clear and huge difference in these frequencies. In some cases, I won't show you. In different mutants, we working with larger population, we ended up with the causal mutation in just one analysis. In that case, we had a smaller population. What we did is to use the identified chromosome. We had different candidate regions showing this SNP frequency ratio that we were waiting for example, this one. We designed, using the different EMS mutation, we designed markers, which are Caspar markers, quite easy to do and cheap to do, and use again same population to find map mutation. And what we found actually is that this mutation was not close to Central American region, but it was in a recombinant region, which was nice. But we didn't identify it, and we did using markers doing analysis of recombinant and so on. We didn't identify any causal mutation in this region. So, but we knew that mutation was there. What we did in that case is to go back to the data and to look at all different mutations, because EMS actually is known to cause base-base transition, but what we know, and some other groups did the same in tomato, we know that we have not only base-base transition, you have also transversions, and you have also indels. And actually, the mutation was identified again by looking at the mutations that were filtered out in the first step, and we identify one T deletion in one gene, and we clearly see here the frequency in the mutant bulk and in the wild type bulk, but is very close to what we are waiting for. And what we did, again, using different mutations as marker, we could, without any doubt, assign the mutation. This is mutant, this is wild type, and you can clearly see that this mutation is responsible for the mutant phenotype. So now we are at this point. This is very recent work. Actually, we did the sequencing in August or September, then the analysis, and we came to that a few weeks ago, and we are at this point now. And actually, the candidate gene that is responsible for the phenotype that you observe is a guanylate binding protein. We don't know much from what I've seen on that role in plants, possibly involved in C cyclic GMP signaling, and this may interfere with other hormone signaling pathway, gibberellin, auxin, ethylene, nitric oxide. And this is the question we are working on. Second one is to be absolutely sure. We know that thin pericap is linked to this problem of cell division. Using the different markers, larger population, we want to be absolutely sure that this delayed ripening is due to the same mutation as the one responsible for that. So this is one example of a strategy that we are using. I will just talk a few minutes about another work that we have done on fruit cuticle. Again, same strategy, going through database, identifying mutants that showed fruit surface appearance different. Glossy mutants, you can see that they are, compared to white type, they are more shiny, and dull mutants. So what you have done, and this is the work of Johan, is to identify a series, we are not working on all of them, of glossy mutants, series of dull mutants, and analyzing these, we could have some ideas of the processes that control fruit shininess, glossiness. And just to give you an example of how interesting it can be for biotech application later, this is, Johan is working very well, but he may have some, forgot some tubes in the fridge, what he did. And this is a white type mutant 1 year after being in the fridge. White type mutant, cuticle affected mutant, and another cuticle affected mutant, but in that case, that mutant still after 1 year showed very good appearance. So this can be quite interesting for looking at cuticle effect on shelf life and so on. But what I want to show you is how we try to identify the underlying genes. So as I said before, this is an outer pericarp, you have a cuticle here, and cuticle is made of two main polyester. One is cutin, the other one waxes with different components, and this is cutin, total cutin load on these different mutants. So you can see that in the glossy mutants we have several cutin deficient mutants. We identified the first one, which actually is a cutin synthase through map-based cloning. And this one, we wanted to go for this BSA NGS mapping strategy. So we have done that, again, a different bulk. In that case, we have been working with 400 or even 600 BC1F2 plant. Again, you can clearly see that the chromosome that carries the mutation, you have a clear difference in the SNP frequencies, narrowed down to different SNPs in one region, 10 to 15 SNPs, and again we could identify the SNP that was present in homozygous mutant line and responsible for mutation. And actually what we saw is that that mutation was a glycine to arginine mutation in that gene, which is GPAT, glycerol phosphate acyl transferase. And this mutation is very close to a domain that is responsible for catalytic activity of the enzyme. And you introduce a charged amino acid next to this domain. And we think this is responsible, and we are working on that to demonstrate that responsible for the loss of activity of GPAT and for the cuticle phenotype. And GPAT is a well-known enzyme that is involved in the synthesis of cutin monomers. So these are just two snapshots of current work that we are doing on these mutants, something that is far from being finished, but I think it demonstrates how we can identify genes we wouldn't have think of before, or new alleles for known genes that might have strong importance for applied aspects in crop breeding, tomato crop breeding. One main advantage of these microtome mutants, this strategy, This is one table in our greenhouse, and you can have one whole population, segregating population on 4 square meters. So that means that you can grow them about year-round in greenhouse. You can have different population, phenotype them, then go for sequencing. And if you are lucky, if it's in a recombinant region and so on, you can straight identify the gene. And you are currently, and we sequence different mutants, these high-low mutants, where yield is double of normal microtome, cracking aspects, vitamin C, and so on. And just one of the last slides, you can further go and use this mutant collection for looking for allelic series in the gene that you identified before. One example for this gene, which is involved in carotenoid biosynthesis in the fruit, we have isolated several missense mutations that do not cause any effect on the probably function of a protein, white type phenotype. And then missense mutation, again, glycine to arginine mutation, strong effect, and you get the tangerine phenotype. The same with this cow mutant. And in that case, you accumulate cis lycopene instead of trans lycopene. So I think now we begin with tomato genome sequence that is available not only for Heinz but also for different cultivars. With all the tools, and we had a very nice tour this morning, the facilities here. I think with genome sequencing, bioinformatics, and so on, we have all the tools, I think, in tomato to use these mutants, but I think it's the same for all plants, crop species in which we can have EMS collection. And next, we can also use, but this has been known for a while, tilling to identify allelic series. So I would like to present you the people in my group who participate in all that work. Lucy is carrying out all the work on the fruit size mutant. Johan, that is here on the Cuticle. And Daniel is somebody that is central to all mutant collection, greenhouse, and so on. And Cécile, JP, and so on, have been working on all this NGS mapping and did a very good job on that with Virginie. And Martin was carrying out all the work on the basic transcription factor that I showed you at the beginning. And of course, we are not working alone, and we have been working with different groups. And I forgot, for example, Ali for metabolome, Ali Ferni from Max Planck Institute in Golm in Germany for metabolome analysis and so on. So I wish to thank you for your attention.
This has been a production of Cornell University, on the web at cornell.edu.